Good morning. Let's see, recording is on. Good. So, quantitative big data again, and today I'm going to talk about data sets and also ground truth references on how you're working with your data. And first of all, we are going to talk about creating data sets. There are also many databases out there with data sets. Going to talk about what is a good data set and how to build your own data sets if you would need that. Then sometimes data sets are not sufficient, so then we can work with augmentation, so modifying existing to be a bit more. And then when we are working with the analysis, we always need to have some baseline. Is my algorithm good or bad? And for that, we need a baseline at least to see what, how good the current algorithm is. As usually, we have a bunch of packages. And if you want to run this presentation notebook, you need to run this one at least before you start doing it. As I said last week, this is a Jupyter notebook. So many of the figures, they are generated using Python code. In the slide, they are hidden, but you can always look at it if you want inspiration for your own visualizations or ways to do things. So the whole source code of the things I have produced within the notebook is available and you can run it. Then there are some references. I'm not going to read it through, but these are interesting reading for those who want to dig deeper. And then there are some augmentation tools that can be used for modifying the data. Honestly, I haven't checked since last year. These tools, they come and go, so it could be that one of them doesn't work anymore, and instead there will be 3 new ones that could be very useful. So actually, I haven't checked out on that one. So first of all, a little bit motivation. So I'm talking about data sets, meaning it's already existing, and one question is, why other people's data? And this is actually, well, a motivated question, but the reason is that existing data actually is already existing. It's also like a benchmark. If your algorithm can perform on the same data set as other people's algorithms are working on, then you can compare also what they're doing. And let's see what other people say about the data. In principle, here are some, at that time when these were screenshots, it was actually tweets, not X's anymore yet. But in principle, what different people are saying is that they get much more out of their machine learning from having well-prepared data than training fancy machine learning models. Of course, you can always tweak and mix around with the models, but the important thing is actually to have data that you can trust and work with. Another example is the slide. Amount of hours lost of sleep because of one is the data set and the other one is algorithms. And if you look at a typical PhD, spend a lot of time on developing their models and algorithms and just a tiny bit on the data. Apparently at Tesla, they spend 3/4 of the time on polishing the data, making sure that they have the good data sets, and in the end, they do 1/4 of the time do modeling. And that is the way how it often is done. Also, this slide is nowadays a couple of years old, so that was before things were happening over on the other side of the pond. Anyway, here is another one. Also a comment that 95% of the time this analysis lead is spending 95% of her time on building tools for scraping, mine, and annotate her data, and finally doing 5% of her time on, say, the machine learning algorithms. So, data is definitely important. It's what we actually want to work with. The question is only how it's to use it. And the important thing is to test your algorithms, you need some data. And first of all, you need to know what to, if you don't know what to expect, then how can you know that your algorithm actually worked? And also, if you just have a few edge cases, you can't know that it actually covers the whole space. So you have to make sure, of course, that the edge cases work, but also that the entire framework is working. And also, if you have these big, say, development houses policy like Facebook, they actually have some iteration where they develop new algorithms every few hours, and you really need to be sure that you can test with confidence that this algorithm actually performs better than the previous ones. And so there he comes. And that, of course, is an important thing, in particular when you work with machine learning. You can, of course, write an algorithm to count cells, but if you want to try to detect subtle changes in the cell structure, or maybe that indicate some cancer, it's not always easy to write an, say, traditional analytical way based on mathematical rules to define this. So that is where we could be using the machine learning. Important is that you can make your data reusable. In particular, if your team working with data, of course it depends on where you are. If you are on the, say, method development side, or if you actually have your own little research project where the data is the central thing. But still, your data can be used by a colleague to get one step further in their project. And often our algorithms, when we start developing them, they are certainly messy because we may not always have a clear idea what it's going to be doing, and then it gets complicated, and typically also when it's messy, complicated, can also be poorly written, and that's, of course, because everything is produced under time pressure. You have to produce your paper, your thesis, and it's not always the code quality that is important. But to be sure that your algorithm is doing what you expect it to, then again, you need to have the data that can work with it. And then by using this recycled data within your lab, within or even on a larger context, can improve your development. When I look for data, I actually ask, so in the beginning, I ask for 3 types of data. The good, perfect case. The bad, the normal thing that you would get. And then, of course, the ugly too, full of noise, artifacts, every nasty thing you can think of. And then this trio, I try to see how well I can perform in these 3 categories. This is, by the way, a new slide I added this morning, so it's not in the script. I may even add a few words in the new notes that I'm updating. But I think this is really, when you start looking at data, to get an idea how to approach it, how to approach the analysis, these are 3 good points to look for. And now switching over to existing data sets, so some famous data sets. We have some general data sets. We have MNIST digits. Well, I'll show the slides later. We have ImageNet, and then for medical, we have BRATS. Of course, there are many more, but these are some traditional ones that come up pretty soon when you start looking for data sets to test your algorithms. So the MNIST digits is a collection of very clean black and white images of handwritten digits, written by I don't know how many people, but it's quite many digits of each category, and they are used for character recognition, and it's used since quite a while as a benchmarking data set. Then we have ImageNet, which is a wider data set. It has also color images, and it's categorized in different classes. You have some kind of hierarchy where you have different categories. You can go down to animals, etc., or people and food. Yeah, a lot of different categories. Another thing is also the bar chart here shows how well different classifiers are working. So this is actually dating back some years, but there was a huge development in how the machine learning was working, and it improved also the performance quite a lot on these ImageNet segmentation tasks. And what these data sets tell is, in principle, you have different categories of people. How they work is very different. And if you come from, say, the experimental side, then you come with domain knowledge. You add your own knowledge into the analysis, the ways how you work. And then when the machine learning era really exploded, it was actually like 15 years after the first neural networks came up. But anyway, it exploded because we had the computational capacity to really do things. Then all of a sudden, the teenage boys in the basement, they could solve classification tasks that needed experts before, just because they had the data. They didn't know anything, and that's a bit a dangerous thing also, that to fully trust what the machine learning is doing. Yesterday I saw a talk by Michael Unser where he showed an example that these networks, they can even hallucinate, invent features in the data if you don't watch out. So you have to still have a critical mind when you work with these models. They do fantastic stuff, but have a critical mind when you use them. Same goes, of course, also with the large language models. Always watch what they're doing with a critical mind. Well, deep learning, does it always win? It's a good performer, but it's not really the point about this lecture. It's more about that we need to have well-labeled, structured, and organized data set to work with as benchmarks and tests for our development. And with that, also getting more confident in what we are saying. Another side of it is working with someone else's data. And Cassie Kosyakov, she wrote quite some nice lines about it. And the first point is that inherited data sets are like inherited toothbrush. You can use them, but it's kind of an act of desperation. So you prefer not to. But the problem is that collecting data is pretty expensive. You can get only so and so many hours on the instrument to collect data. You can only have so and so many questionnaires to ask people because you have to evaluate the questionnaires afterwards. So it's not easy to get the data, and sometimes it's the whole purpose of a master project or a PhD project to collect the data. So it's quite a lot of effort involved collecting data. But on the other hand, inherited data, it's easier to get because it's already there. But you need to trust it, and that is where the problem lies. Another aspect of data sets is also the ethics. And some point here is, first of all, transparency, how it was produced, and also if it can be reproduced. Privacy and confidentiality, in particular if you work with medical data sets, you really need to make sure that they are anonymized and that cannot be connected to any individual before you can use it and distribute it. Another thing is also fairness and bias distribution. It's easy to collect maybe easy to collect a certain type of data, but it can also bias your conclusions if you have a lot of data in one direction. And integrity and avoidance of data manipulation. Well, if you do some kind of modification when you prepare your data, then you have to make sure that it doesn't actually affect what you want to say with the data. And of course also responsible use and governance again comes back to the problem in particular with the medical data. You can't distribute data in any way, so you have to make sure that it's under controlled circumstances. And some best practices. First of all, you have to obtain informed consent. So if you have patients, you have to have the signature that this data is actually allowed to be used in some research context, probably in the context of some you probably had that yourself in context. Maybe you had a surgery and then you have to sign, is it allowed to use parts of this information that we get from your body essentially in some research context, and then you have to sign that kind of paper. There are also the use of ethical review committees. Again, that is depending on the experiment. Also at PSI, when we do experiments that involve patient samples, then we also need to go through the ethical committee. If you have animal experiments, you also have to go through the ethical committee to be allowed to perform the experiment at all. Also, make sure that the data is secure, that that only the right people can have access to it. An important general thing is to promote open and fair data. I'm going to fair on the next slide. And then we have you have to audit that there is no bias in the data, and also that the data is fair in different ways. And the last point, document everything, is extremely important. Without documentation, the data is not worth anything. Because if you just have a bunch of images, or whatever data you have, and don't have a clue the experimental conditions for each sequence of images, you don't know, if you don't know anything of how the data was acquired, you don't really have the full story, and you can't do any investigation with it. Of course, you can use it as images as such, but you can't reproduce anything using that kind of data. The FAIR principle is something that was introduced in 2016 by Wilkinson et al. And there are 4 points that are very important when you talk about FAIR data. First of all, it must be possible to find the data. And today, very many data sets get an DOI, so digital object identifier. And with that, there should be a pointer to where you can find the data. Accessible is the next point, and So you can find it, you know where it is, but you also should be able to retrieve it. And then the next one, interoperable, is maybe a strange one, sounds strange, but it's about having the tools that allows you to actually work with the data. One thing is you can have terabytes of data in your hard drive, but you can't work with it. And the last one is reusable. If you can make data reusable, it's again a question how well documented the experiment conditions were, or the acquisition conditions. And that's actually a thing where I myself sometimes have my doubt about the FAIR principle. The first three, they can be technically solved, but the reusability depends on the one who uploads the data to make the right kind of description, what it actually is. A bunch of imaging doesn't help to make it reusable. There are some initiatives. One is PAN-OSC, that's for the large-scale facilities. We have this association that provides this open science cloud for research data within photon and neutron sciences. I also collected on the class webpage a little collection of data sets that I have found that I think could be useful. And if you go on the webpage, you have the datasets tab here, and then there is a list. I added a few this year, so there are some more. And some of them, they are fully open. Other ones, they are just a list of pointers, and some you have to pay for. Other ones, you or have some kind of subscription to get. And other ones, yeah, they're completely closed, but yeah, that depends a little bit. So, for example, the IEEE data port, you can get access to some of it. You get a bit more if you're a member in IEEE. Google data search engine, it's actually just, I think it's data.google.com. There you can search for data sets. It's not very well categorized, but you get lots of categories. Then there are some open medical image repositories as well, and so on. A big place, yes?
Is it possible to get free access to some of the data with our university email address?
In some cases, probably yes. I don't know, actually. It's so different between different databases. I think you should go through, maybe through the library and see if there is some dataset collection portal or something. Another one, which is dataset repository, is Zenodo. I haven't tried to search datasets in Zenodo, but you can upload, everybody can upload up to 50 gigabytes per dataset with some descriptors. And you can also upload software if you want there. So the software I develop, I have a DOI through Zenodo. And so that is a way if you also want to yourself to upload datasets. There are other dataset services like Mendeley has dataset service. So many of the publishers, they also have some dataset services where you can upload data, mostly up to 10 gigabytes. But and if you want larger datasets, you have to pay for the space you need. For medical imaging, we have the BRATS dataset, which is segmentation with the purpose of segmenting tumors with different modalities of images. So we have MRI and X-ray tomography. And then all these images, they are labeled with different tumors. Actually, reminds me, the lecture yesterday, that one of these machine learning reconstruction algorithms was able to reconstruct the anatomy of the brain, and it forgot to reconstruct the tumors. So you had a nice sound brain after the reconstruction. So somehow it generalized it in the wrong way. It was kind of interesting to see that it does it that way. And of course, they needed to find solutions on how to handle that. So what makes a good data set? First of all, we should have pretty large amount to get the statistics. Also diversity, all the corner cases, everything in the middle should be covered. And ideally, well, it depends on the purpose of the data set, also annotations, what each image or sub-data set actually meant, or regions in the image meant. So that means, first of all, lots of images. In particular, if you have a complicated task, finding something in the middle of a lot of chaotic background, very subtle differences. The problem is, for example, if you have a lung tumor, the contrast range of it is about the same as the normal lung tissue. The thing is only that it's in the wrong position, and that is kind of the indication that where it should be. Very typical is also that you can have class imbalance, and that is when, well, the normal case is people are healthy, but there are few who have some kind of disease or problem, and you have to balance this in the data set too. But any data that you can collect is of course good, but the more data you can collect, the better it is. But the cost of getting it increases. Diversity is also what the data in the wild really looks like, and then we more or less come back to the good, the bad, and the ugly. We have plenty of scanners, reconstruction algorithms that perform in different ways. We have also different acquisition conditions which then have an impact on the noise levels, different illumination, also orientation of the sample or patient, color variations. So there are very many different things that can vary in the data. Of course, you have to be within, say, the natural range, but some things can always step aside the main line. And then, of course, you also need a lot of examples from different categories. And, well, here is one example of kind of an outlier. You only have one case of male breast cancer, so it's very hard to identify what that really looks like, and training from that. And if you only have one single patient among thousands, millions of healthy ones, it's not so easy to really find something that can pinpoint this case. And of course, we need meaningful labels of what you actually see. And of course, you have to have a clear task or question when you start doing your labeling. What is it you want to label? And there should be no ambiguity in what it actually is. Should also be non-obvious. I mean, if the image you see is yellow and you write it's yellow, it doesn't help very much. Also, it should be able to derive it from the image alone. So, for example, if you add a patient's health insurance, it doesn't actually bring much to the analysis of the image. It can help you for some other statistical evaluation, but for the analysis of the image, it doesn't help you. And ideally, you should be able to quantify it in some way. And depending on your task, you can also have different purposes of your data sets. And the typical image processing task we have is classification, regression, segmentation, detection, maybe something else. And if we look at first the classification, is to look at an image, and that's actually what is happening in everyday use on your phone. You can categorize for different people, classify different people. You can also classify for different items, things. And they should also be non-ordered, so there shouldn't be a specific order in these categories. So the classical one is cat versus dog, or healthy versus cancer, etc. So you just take an image and say, this is what's on it. You can also, for me, I'm always looking for some kind of food, and then I say, this kind of food, and I get that in my pictures. And here we have a classification example. Again, coming back to the MNIST. And this pink band here is, well, just some kind of information actually related to the TensorFlow on the Mac. Always get that kind of things, but it doesn't actually something you need to bother much about if you have a Mac. Anyway, so here we got some classification example. We show the digits we have here, and they're not perfectly black and white actually. They're a little bit grayscale in them, some anti-aliasing along the edges. And regression is when you have an image and want to map it to some kind of value. The classic example is the value of a house. You make a picture of a house, and then there is a big database of different houses, and they are associated with values. And through that, you can do a regression. Just from the picture, it can tell you that this house is probably worth 1 million, or this house or shed is only like 100 francs. Another thing we can also do with regression is having satellite images, looking for the weather conditions and telling, for example, the risk of a hurricane. That could also be a regression task. Here's another example. I think it's a Kaggle competition to tell the age of the child when you see an X-ray of their hand. So all these have the age of the child associated with the image, and then you can just bring in a new hand image, and it can tell you how old the child is. Segmentation is another task, which is also pretty commonly happening today. If you just click on your phone, it marks out there is the person, and that is a typical task that's also done within the image processing. And typical examples, if again is a tumor, if there is a tumor in the lung image, or where the streets are in some satellite images. Again, the cats and dogs. Cats and dogs are very popular in these tasks for some reason. So that would be, in principle, you have this picture of many cats and dogs, and it should tell you which ones are what. Today, the light in the room is a bit bright, but here you have some microscope images of cells. And if you look in a darker room, you would see that actually there is a labeling of these cells in the image. And that is also some Kaggle exercise. If you want to look more into that, just follow the link on the slide. Detection is also to predict if something appear in the object, and maybe even just putting a bounding box on it. Could be cars on a street moving by. And it could also be that you have multiple objects that overlap each other. So here we have opaque regions in X-rays, and then it marks out boxes where you have the different opacities in different regions. And yeah, there are many other applications of data sets. Next week, I'm going to talk about image enhancement, and there we come into the topic of removing noise from images, and also looking into super resolution is another task where you can use these data. Typically, when you look at super resolution, you would probably downsample an image, and then you try to reconstruct what was the true resolution, and you can use the ground truth for that. To build your own data sets is quite a task. So first of all, it's very time consuming, and it has improved, but there is not actually a lot of great tools for it. And also building is very problem specific, and that is where your own knowledge is coming in into the task. There are different ways to approach it. First one is code-free, so you don't program at all. But it's more, the first one is actually just how to organize your data. You can have folders where you have different categories. So you have one folder of cats, one folder of dogs, third folder of humans, and so on. For the regression, you organize it in principle with a spreadsheet where you have one column with the file name, and next column is the category what you want to, or the value you want to associate it with. Actually, you can do that for the classification as well, that you have a list, and then in the list you tell the category for each image. For segmentation and object detection, you can take some kind of paint tool, and like Fiji or any other painting tool, and draw a region. So if you have a grayscale image, you can just paint with red, for example, and then you see the regions that are relevant for something. Then there are several tools for data labeling. There are different ways you can do it. One that is a little bit fun is maybe the tint of a brain MRI. So you just swipe, is it a tumor, is it not a tumor? That is one way, and it's relatively quick. So you have to have tools that, if you have many images, you have to have tools that make life easier to work with. And here is an example where I actually had an image that I wanted to mark up. Actually, I did it for the lecture purposes. In the radiographs that we produce at our beamlines, we get a lot of white spots. So these are outliers, and they are very annoying in the next steps of the processing. So we want to remove them. And I'm working on different algorithms to remove these spots without affecting the rest of the image. So what I did, I took the original image, 2K by 2K, and zoomed in and started painting. And painting, and painting. 8 hours later, I was finished. That was pretty mind-numbing, but in the end, I had a nice example for testing with. So this is an example how much time it can take. Other approaches would be to use, say, a baseline algorithm to mark up, but you can't trust that the baseline is really good. So then we come back to the human labeling again. You can also do simulations. So you build up a model of the scenery you want to look at, or have a very nice, clean images, and those you then afterwards destroy or corrupt in some way that you have all the artifacts and the noise, etc. on them. But if you do a pure simulation, you have geometric models and or template models. Many years ago, there was this project about building a brain atlas that, that more or less had a growable brain shape, and then they could put in different sizes and distortions in that model. Some examples. One is that you actually have real data, and then you have the accurate ground truth, and then from the real data you do some modifications. For example, in tomography, you could have a very, very nice tomography data set, and then afterward you do the synthetic forward projection, producing the projections, and then you have your projections that you can apply in different reconstruction algorithms, and then you can see how well it actually resembles the original. One very nice example of the simulation, synthetic ground truth, was presented at this conference. I think meanwhile it's also a full paper there, where they had some pore detection in wells or casts. So they produced really a sample of some aluminium, introduced some realistic holes in it, and then they did a forward projection with Monte Carlo simulation, I think, and then they did the whole reconstruction, segmentation, to see if they actually could do reproduce the whole process. So this is a very nice paper if you want to see that workflow. And some problems with the data set is that I already mentioned the imbalance, also too few examples. Maybe it's too homogeneous. You only have very narrow examples, very similar examples, and so problems like that. But that also gives a problem when you want to check the performance of your algorithms that you're working with. One problem is also bias. This is a classic, is always mentioned in the case of bias in image recognition. And well, Google apparently had a categorization of two people. They were categorized as gorillas, and some got pretty upset and said, you up, and his friends are not gorillas. And it's apparently a true story. What they did to solve it, they removed the category gorillas and everything was fine. So that's a quick fix, and I think they fixed that within a few minutes after they got the notice. But the best way to avoid biasing is to have more and more data, and with more variations. So for example, you have different categories here with eyeglasses, bangs, pointy noses, oval faces, etc., to get a lot of categories out of the data. And that is where, for example, this IBM diverse data set comes in play. Then we have this labeling and data sets. So come back to the numbers from the MNIST. Now I have reduced it only to the categories 1, 2, 3. We have these 3 data sets. And what did I want to say here? Yeah. In principle, we have this limited data set. Yeah. Another thing with the limited data is that we have only few points or limited amount of images. The unbalanced data is really a big problem. It's in particular for the outliers, is that we have a very small representation of a certain category. And if the category is far away from the distribution of the rest, you can handle it. You can put the threshold somewhere around here, and you have separated them, even though it's very unbalanced. However, if this class is merged within the main distribution, then you have a problem. You can't really detect it. You can see here the change of the observed histogram is just a tiny bump, and you can't really distinguish it. And this is where the problem with unbalanced data comes in. And then, of course, you have little or missing training data is, of course, also a problem for your data sets. And, well, soon the bell will ring, so I wait with augmentation to after the break.